Okay, where were we? So you have these different fragments of DNA that are differ in length by one base, and they are each one of those is fluorescently labeled, and the base at the 3' end, the different fluorescent labels are determined by which base is at the 3' end. So you need to find a way of detecting these things separately. So you do this by gel electrophoresis, and in the middle of the top is a, I'm going to have to do this again, I think. Nope, yes I do. There. Okay. Gel electrophoresis using a capillary tube. Capillary tube is a glass tube with a very small diameter, internal diameter. And you load all these molecules at the top, and you turn on the electricity, and they migrate downwards, and the small ones migrate faster. And at the bottom of the tube, they are detected by a laser, and the fluorescence is detected. As they leave the bottom of the tube, their fluorescence is detected. And what you end up at the bottom of this figure is it's basically a graph. The x-axis is time. This is the time at which these molecules elute, and the y-axis is fluorescence intensity, but it's color-coded. So there. Then the upper right is shown how this is done. This is a 48 capillary. You do this to sequence a genome, or you need to scale this up. Each one of these reactions, you can see in that picture, is giving us reliable sequence out to around maybe 600 bases. Maybe it could go to 700 bases. But if you want to do an entire genome, you have to scale this up. So what is done in this cartoon is a 48 capillary array. You have a machine that does 48 capillaries simultaneously. In fact, the ones I'm aware of do 96 capillaries simultaneously, and they're loaded robotically, and they do the electrophoresis for, I think, about 3 hours, and they record the information, and then they flush out the stuff that's in the middle of that tube, and then they replace it and reload it robotically. And they do that every 3 or 4 hours, all day and all night. And then they have a room full of these machines, and then they have laboratories around the world, and that's how you sequence a human genome. Okay, so say we're doing a bacterial genome, and we end up with a genome sequence. So here is a sequence of DNA that is meant to be raw sequence. It's just a telephone book of A's, G's, C's, and T's. The top strand is one strand, the bottom strand is the complement of it. Oops. So the question is, how do you find the genes? And the answer is there are various clues. So I'll tell you the answer. I'll tell you where it is. I picked a very small gene. I'll tell you the answer. The start codon for this gene is that ATG, and the stop codon is this TAA. Okay, and the protein sequence in single letter code is this. There's a methionine. This happens to be an arginine, then a serine, and a leucine, and so forth, down to a serine at the other end. That is the gene. I picked a very small gene so I could fit this on one line. This is a very small gene. A typical gene is about a kilobase. This is, I can't remember how much, 20, 40, 60, 80, 100, it's 140 bases. So it's 10 times less than a typical gene. Okay, but say you don't know this. This is the goal. Say you don't know this. What do? Well, you ask the machine to do conceptual translation. Conceptual translation. So what that means is that the computer is being asked to take these DNA sequences and predict protein sequences from them. So there you have it. The computer is taking the bases in triplets and just inferring the correct amino acid for each triplet, moving down in 3 bases at a time, and making the inference about which protein would be, which amino acid would be encoded in each one. It's doing it in all 3 conceptual reading frames. So it could, on the extreme left here, the TAA, it's saying, okay, if that's a codon, well, what does it encode? Well, it happens to be a stop codon. The next 3 bases, TGG, happens to be a W, which is a tryptophan, and so on. But what if it's not starting on TAA? What if it's starting on AAT? Starting in that reading frame, AAT would encode an N, which happens to be, help me, glutamine, And the next one would be this GGA would be a glycine, and so forth. What if it starts on the third reading frame? That ATG, that's a methionine, and then the next one is a GAT. That's a D, which is who knows what, aspartic acid. This computer is translating this sequence in all three possible reading frames. But you can also worry that the gene might go the other direction. So there's 3 more reading frames reading in the opposite direction. The computer is happy to translate all 6 reading frames, putting in whatever the sense code, whatever the amino acid is. And you'll see here, there's a number at the upper left is a number sign, and another number, that's a stop codon, and that's a stop codon. And this asterisk is a different stop codon, and that plus sign is the third stop codon. And you have a stop codon here, and here, and here, and here. None of which are real stop codons that are really used by ribosomes to really stop translation. They are conceptual stop codons. At this end here, at the 3' end of this gene, is the real stop codon that ribosomes actually do use to really stop translation. The others are conceptual stop codons. On the bottom strand, we have here a plus on the right-hand side, a plus, and we have too many over here. There's a number symbol here, and here, and here. So those are also conceptual stop codons. So what you can do is look for regions of DNA that are devoid of stop codons. So, for example, in this gene, you see these are obviously have to be sense codons, and followed by at the end is the real stop codon, is really used by the ribosome. Okay. This is a very small gene. Imagine a gene that's 10 times bigger. There would be a conspicuous lack of stop codons in that gene. All the other 5 reading frames, the stop codons are sprinkled randomly, and there's fairly large numbers of them. That's the key. You look for a long sequence of sense codons with a conspicuous absence of nonsense codons. And that's what we see in this cartoon here. In reading frame 1, what they call reading frame 1, there is a start codon here and no stop codons, and then at the other end, way down here, is a stop codon. The other reading frames don't have any uninterrupted string of sense codons of anything like the same size. So there's little tiny strings of sense codons. It could be an ATG, an AUG here, and then a stop codon here, and so on. And in the bottom strand, the same thing. There can be short strings of sense codons, but they're too short. They're unlikely to be real genes. So once again, this conspicuously long string of sense codons with no stop codons is already some evidence for the existence of a gene, and the gene would be in that reading frame. So we call this an open reading frame because it's open, because it doesn't have stop codons that would, I guess you could say, close the reading frame. Anyway, so that's one kind of evidence that the real gene might start, as shown in the cartoon, at the left end of this green line and end at the right end of that green line. That's one kind of evidence. There should also be, if this is an operon, you can imagine that the stop codon of this gene lies close to the start codon of the next downstream gene, and the same thing at the left end. The stop codon of the upstream gene would lie close to the start codon of the downstream gene. That's more evidence. There should also be ribosome binding sites. The real start codons, here's the real start codon, might, if you're lucky, might have a decent ribosome binding site. Methionines are also found in the middles of proteins, and they don't have ribosome binding sites. So here's a methionine within the middle of this gene, and there's no ribosome binding site for that because it's not needed. So if you have a methionine preceded by a ribosome binding site, that's evidence that is an initiation methionine codon. I think that's it. But what's even more useful than any of this is you take this gene, this amino acid sequence, this is one single letter code, M, R, S, L, 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 M, G, and so on, all the way down, and you feed that to a computer, and you ask the computer to look for, to search in protein sequence databases for sequences that are similar. And I did that here. I took that sequence and I sent it off to the computer and so this is the sequence that I have submitted, and these are sequences that it found that it thinks are similar. Okay. Now the sequence that I fed it is already in the database, so of course you're going to get that sequence back again. It's going to be the closest match is going to be the very same sequence that you already gave it. But then it finds other, so we kind of ignore that second line because it's just the very same gene. Then we have other sequences that are not identical, but they're similar to each other and similar to the sequence that we're trying to study. The existence of these genes in DNA sequence databases is evidence that that this is the real gene. You can feed the computer all these other sequences, just ignore all the stop codons and just give them all 6 reading frames, and it'll find no sequence similarities in any of the other reading frames. It'll find this in this reading frame. It'll find no homologous protein sequences from the other 5 reading frames, and that's pretty nice evidence that this is the real reading frame, and that this is the real start codon. Sometimes it's a little bit tricky to know the real start codon because this methionine, maybe it's the real start codon. Well, this one has a ribosome binding site, this one doesn't. So you can kind of make a, okay, you sort of make an argument that that's the right start codon. And this start codon aligns with other start codons. Sometimes then they differ a little bit. Okay, you can search the computer data. Sometimes it could be that something is known about these genes. If you think this is a gene and you think this is something you'd like to study, and you say, well, what does it do? Well, you can read about what these proteins do and make an inference that maybe your protein has similar properties. Now, this is the next slide. This is from a textbook. There is imagine that you use this protein sequence to search a protein database, and it comes up with similar proteins that have similar sequences, and maybe some of those proteins have been studied, and they are known to be, or at least one of them that's been studied is known to be a dihydrolipoamide dehydrogenase. Now, it doesn't really matter what that is, but the point is that someone has concluded that one of these proteins has that activity. Furthermore, that protein might have a binding site for a cofactor, FAD, might have another binding site for a pyrimidine redox active site, might have another binding site for NAD. And you can make inferences that maybe your protein has all those same properties. That's very powerful. So you can do this for individual genes, or you can do this for entire genomes. And that's what you get here. This is an entire genome. You can, like I say, you can search the protein databases, protein sequence databases with single genes, or you just feed the whole genome to the computer, and it will come back and say this is what it thinks is encoded by that genome. And what we have here, I think the outer circle is genes that are read clockwise, and the inner circle, the next circle, is genes that are read counterclockwise. The third circle are insertion sequence elements, and so on and so forth. But the most important thing is over here, that by studying where the genes are and what they encode and what they resemble, it's possible to make inferences about that certain numbers of genes here may have roles in synthesizing amino acids. These genes, I'm looking on the left-hand side, is key. Some genes are predicted to have roles in DNA synthesis. Some may be involved in the cell envelope, and so on and so forth. So you can learn a lot about what genes are present and what they do by doing a genome sequence. Okay, this is showing exactly the same thing. This is a hypothetical, I don't know which genome this is, some genome of some bacterium, and from that sequence you can make infer that it must have a permease that enables uptake of glutamine, and another permease for uptake of dipeptides, and another permease for uptake of oligopeptides, another permease for something else, and all these permeases, all these pathways, and so on. And you can make that inference just from knowing the genome sequence. You can also make an inference about, what from the genome sequence does it have genes that look like they would encode toxins, or adhesins, or something else that might be important for a pathogen. You can, if you see genes that look like shigatoxin genes, they probably are shigatoxin genes, and this organism probably makes shigatoxin. So that's all available from a genome sequence. Okay. this is getting toward the end of this section. It's sort of saying that what might seem awfully obvious, that on the left-hand side is a graph of each dot here represents one genome. And it's saying here that on the left end of this would be a small genome, maybe a half megabase genome. And this is the total number of open reading frames, i.e. the total number of genes in that genome. And so if you have a small genome, if you have a half megabase genome, perhaps you have 500 genes. At the other extreme end, if you have a 9 megabase genome, that's a big genome for bacteria, you might have 8,000 genes. Okay, so what we're saying is that the bigger the genome, the more numerous the genes are. And you might go, well, of course. But it didn't have to be that way. You know that in your genome, most of the DNA is junk. And that's not the case in bacteria. I mean, there's junk DNA, but even the junk DNA encodes something. Most of the DNA encodes something. There isn't large amounts of just what seem to be random sequence. Most of the DNA is encoding a protein or encoding an RNA or something like that. There's one off, this is off the line. This one here is relatively few genes per megabase. That happens to be the Mycobacterium leprae, which causes leprosy. And it is an outlier. It seems to have a lot of junk DNA, but it is the exception. Okay, on the right-hand side, this to me always gives me a headache. It says here, the x-axis is total number of genes in the genome. So on the left side of this x-axis would be a small genome, on the right side a big genome. And the y-axis is relative percent of genes that encode a particular class of functions. Well, maybe it'll become clearer soon. The red line, is saying that these genes are involved in translation. And the idea is that the absolute number of genes involved in translation is pretty similar from small genomes to big genomes. So the proportion of genes that are devoted to translation is high in small genomes and low in big genomes. The number is pretty much the same for all bacteria. So if you have a small genome, then a large fraction of those genes are involved in translation. Same thing for DNA replication, so and so on. At the extreme other end, let's look at this yellow line here. Signal transduction, the ability to detect environmental stimuli and make an appropriate response. Transcription, I guess that's probably pretty much the same thing. In small genomes, it seems like there's relatively few genes that are devoted to detecting stimuli and making responses. These small genomes are generally found in parasites, parasitic bacteria that parasitize a host. So in those kinds of bacteria have a fairly constant environment. They don't have a lot of novel stimuli that they have to respond to. At the other extreme end, if you have a 9 megabase genome, these tend to be like a soil bacterium that has all sorts of novel stimuli, new opportunities to eat something, or a new threat, or a new something that it has to be able to detect and respond to. So they have big genomes, and by far they have a bigger fraction of their genomes is devoted to detecting signals and responding to signal transduction. And so there. So the idea was that these soil bacteria need to think. They need to detect and respond. They have bigger brains, question mark? That's the idea. All right, finally, this is a technology already a little bit, I won't say obsolete, but venerable. If you want to identify every gene that responds to a certain stimulus, this is a technology for doing that. So say you have a genome that has 4,000 genes. You want to say, if I treat these bacteria with acid pH or some other stimulus, I want to know which genes are altered in their expression by that stimulus. So what one can do is to grow a culture of bacteria at neutral pH and grow the same bacterium at acid pH. Purify the RNAs from both of these, okay? And then you convert these to You make these fluorescent with a green. So in this cartoon here, it's a green fluorescent molecule. So these are now fluorescent green, and these RNAs are now fluorescent red. It turns out the simplest way to do that is to convert them to DNA, but why don't we just ignore that and just imagine that these are RNAs that have been made fluorescent. Okay, two different colors. You combine them, and then you add them and you hybridize them to it's a microscope slide. Imagine an ordinary microscope slide with hundreds of thousands of DNA fragments spotted onto it. This is showing, a few hundred, but in fact you can get these slides that have a few 100,000 different spots of DNA. So here's a blow-up of that, and it's showing here 16 spots that correspond to 16 different genes on this genome. And these are single-stranded DNA fragments that can be hybridized to this these probes. It occurs to me that these are actually, the cartoon shows double-stranded molecules, which is probably not right, is it? There should be single-stranded RNA molecules. Okay, so you have these fluorescently labeled single-stranded nucleic acids that were derived from these bacteria grown at neutral pH or at acid pH. Now you pool them and you use them to hybridize this array. And so for example, this second gene over on the top row, the green one, it appears to be expressed. It's green. It means that the DNA, that RNA from pH 7 is more abundant. And there's, in the cartoon, I guess there'd be, the gene is turned off at pH 4.5. So for that particular gene, there's a lot of the RNA at pH 7, and there's little or no of the RNA at pH 4.5. So when you can pool them, these RNAs can hybridize to that gene, and the other one does not. Over here, the red one's the opposite. So that gene is expressed at acid pH but not neutral pH. Let's back up. The green gene here, these green spots, those are expressed at neutral pH but not acid pH. The red ones are expressed at acid pH but not neutral pH. And most of the genes will be yellow because they are hybridized by both. Both probes hybridize the genes in yellow, which will be the most. Most of them won't matter. But a few genes, maybe, oh, what just happened here? Let's go down. Most of these genes, so you might find 50 or 100 genes that are preferentially expressed at acid pH. So the idea is you've identified every gene that responds to that stimuli, whether it's increased or decreased. So that's a very powerful technique. It's a little bit obsolete now because what one can do is isolate the RNA at this pH and this pH and just give it to a sequence facility, and they can just sequence 10 million of these RNAs. And for any one gene, you might get more RNA sequences from one and less from the other, and you can make inferences about gene expression from that. And that's the cool new way of quantitating the expression of every gene in an organism. And it requires extremely high throughput DNA sequencing. It's not the kind of sequencing we just saw. It's a newer level of DNA sequencing that is becoming pretty common. That's about it for this. So I'm going to stop this and save it.